What if we could rewrite the story for pancreatic cancer? What if instead of dread, we had detection? That's what we're diving into today.
Sounds intriguing.
It is a potential breakthrough from Oregon Health and Science University.
Oh, okay, and what have they discovered?
A new blood test called PAC-MAN.
PAC-MAN.
Yeah,
Catchy name. What does it stand for?
short for protease-activated magnetic nanosensor.
Ah, now that you mention it, I think you sent over some research about that.
Exactly. We've got clinical trial data, the science behind it, the whole 9 yards, because, well, you deserve to know if this could really change the game. See, pancreatic cancer, it's notorious for being diagnosed late.
Yeah, that's right. When treatment options are unfortunately very limited.
And that late diagnosis, it makes it one of the deadliest cancers out there. So a test that could reliably detect it early, well, that would be huge. But before we get ahead of ourselves, let's talk about what we're up against. Currently, doctors use a blood test that measures a protein called CA19-9.
Right, CA19-9. It's helpful for monitoring the disease, but not so reliable for early detection.
Okay, so what's the main issue with CA19-9 when it comes to early detection?
Well, for one thing, it can give false positives, which, leads to a lot of unnecessary stress and procedures. But more importantly, CA19-9 levels often don't rise until the cancer is already pretty advanced.
And by then, it might be too late. So this new Pac-Man test, it takes a totally different approach, right? How so?
Absolutely. It focuses on the activity of certain enzymes in the blood called proteases.
Proteases.
Mhm.
Okay, remind me what proteases are again.
Think of them as like tiny molecular scissors
Okay.
that break down proteins. And certain proteases are known to be particularly active in helping cancers grow and spread.
So if we can measure their activity, we might be able to catch cancer in those early stages before it becomes a serious problem.
That's the idea. Researchers designed a special peptide probe attached to a tiny magnetic nanoparticle. When this probe bumps into active proteases in the blood, it gets cleaved, releasing a fluorescent signal.
Like a little red flag going up.
Precisely.
Hey, there might be cancer here. So how many of these probes did they create, though?
Well, they didn't just create one, they actually meticulously tested 12 different peptide probes to see which ones were most effective at differentiating between blood samples from people with and without pancreatic cancer.
12. That's quite a lot. Why so many?
They wanted to be absolutely sure they had the most accurate probe possible for this test. You see, through meticulous testing and analysis, they narrowed it down to a panel of 6 and then ultimately zeroed in on a single probe, probe 1.
So probe 1, that's the star of the show. But why get through all that trouble if you're just going to focus on one at the end of the day?
Because what they found with probe 1 is truly remarkable. It's incredibly sensitive to a specific protease called MMP2.
MMP2. That rings a bell. Doesn't MMP2 play a major role in tumor growth and spread?
Precisely. It's one of those proteases that helps cancer break free and invade other tissues. And Proban1 seems to be tapping into that process very, very effectively.
Okay, so this all sounds very promising in theory, but how does it actually translate into real-world results? Did they test this on actual patients?
They did. They conducted a blinded validation study with over 200 participants. And the results, especially when PAC-MAN was used in combination with the CA19-9 test, were incredibly promising.
Hold on, they combined the new test with the existing one. What was the rationale behind that?
Well, sometimes combining different tests can provide even greater accuracy, you see. Think of it like getting a second opinion from a specialist.
Okay, 2 tests are better than one. Got it. So tell me, what were the actual results?
They achieved 100% specificity.
Wait, 100%. Does that mean no false positives?
Exactly. Every single person identified as having pancreatic cancer actually had it.
Wow, that's incredible. But what about the sensitivity? How good is it at actually catching real cases of the cancer?
The combined test had 84% sensitivity, meaning it correctly identified 84% of the people who had pancreatic cancer.
So it's not perfect, but it's definitely a step in the right direction.
Absolutely. And there's another exciting finding from this study. PAC-MAN seems to be particularly good at detecting early-stage cancers.
So catching those cancers when treatment is most likely to be successful, this really does have the potential to change the way we think about and approach pancreatic cancer, doesn't it?
It really does, but of course there are still some questions that need to be answered.
Like what? What are the next steps for this research?
Well, first and foremost, more research is needed, of course. Larger, longer-term studies are crucial to confirm these findings and see how the test performs in, real-world settings.
That makes sense. But what about the cost and accessibility? If this test proves successful, will it be widely available? Would it be affordable?
That's something they're definitely working on. One of the goals of this research was to develop a test that's relatively inexpensive and easy to administer. You see, it's a simple blood test, which makes it much easier to implement than more invasive procedures.
Okay, so there's definitely a lot to be optimistic about here, but let's play devil's advocate for a moment. What are some potential downsides or challenges that we should be thinking about?
Well, one challenge is ensuring that the test is truly specific to pancreatic cancer. We don't want it to flag other conditions and cause a lot of unnecessary anxiety or interventions. More research is needed to confirm its specificity in, well, larger and more diverse populations.
Good point. And even with a perfect test, there's still the question of treatment, right? How do you effectively treat early-stage pancreatic cancer?
You're absolutely right. Early detection is only the first step. We need to ensure that we have the right treatments in place to really capitalize on that early diagnosis.
So it's a two-pronged approach. We need better detection and better treatments to truly move the needle on pancreatic cancer. But if PAC-MAN delivers on its promise, this could be a game changer.
Absolutely.
And speaking of changing the game, this leads to a really intriguing question.
Go on.
Could PAC-MAN eventually be used to screen for pancreatic cancer in the general population? What do you think?
Now, that is a question that deserves a deep dive of its own. Population-level screening for pancreatic cancer. It's fascinating to think about, isn't it? Imagine a world where we're proactively searching for this disease, potentially catching it before symptoms even emerge.
It's definitely a powerful idea, but I can't help but wonder about the implications. Wouldn't widespread screening lead to a significant number of false positives? I mean, we'd be talking about potentially causing unnecessary anxiety, subjecting people to additional tests, maybe even invasive procedures when they don't even actually have cancer.
You raise a very important point. The issue of false positives is a crucial consideration when evaluating the feasibility of any screening program, especially on a large scale. But before we dive headfirst into the complexities of population-level screening, I think it's helpful to step back and look at the historical context of using protease activity to detect cancer.
Okay, so let's rewind the clock a bit. This whole idea of using proteases as a sort of red flag for cancer, it's not entirely new, is it?
You're absolutely right. The concept itself isn't entirely novel. Scientists have been investigating the connection between proteases and cancer for decades now.
Really? Wow, I have to admit, I was under the impression that this PAC-MAN test was a completely groundbreaking discovery.
It is, in terms of its accuracy and the specific technology it employs, but the underlying principle that's been around for a while, it has its roots in earlier research. Let me give you a bit of history. Back in the 1980s, scientists discovered that certain proteases, specifically a family called matrix metalloproteinases, or MMPs for short, play a crucial role in cancer invasion and metastasis.
MMPs, hold on, those are those molecular scissors we talked about earlier, right? The ones that help cancer cells break free and spread to other parts of the body?
Precisely. They essentially break down the extracellular matrix, which you can think of as the scaffolding that holds tissues together. And by degrading this matrix, they allow cancer cells to move more freely and invade surrounding tissues.
So I'm guessing scientists thought, okay, if these MMPs are so active in cancer, maybe we can detect them in the blood and use that as a way to diagnose the disease. Is that kind of how it went?
That was the hope. And there have been numerous attempts to develop blood tests based on MMPs and other proteases over the years, but these earlier attempts faced significant challenges.
What kind of challenges are we talking about here? What made it so difficult to create a reliable test?
Well, for one thing, measuring protease activity is a lot trickier than it sounds. It's not just about detecting their presence or absence in the blood. It's about understanding how active they are. And those earlier tests, they weren't always very sensitive or specific.
So essentially, they might have missed some cancers altogether, or they might have generated a lot of those false positives that we were talking about earlier.
Exactly. And to complicate matters further, there's a natural variation in protease levels from person to person. This made it incredibly challenging to establish a clear-cut threshold for what's considered abnormal. It's like trying to hit a moving target.
So it sounds like PAC-MAN has managed to overcome some of these hurdles by zeroing in on a very specific protease, MMP2, and by using this very clever magnetic nanoparticle technology.
Precisely. And I think it's important to emphasize that the researchers didn't just stumble upon MMP2 randomly. They meticulously tested a whole panel of probes before identifying probe 1, the one that specifically targets MMP2, as the most accurate one for the job.
Right, they tested 12 different probes before narrowing it down to probe 1, if I recall correctly. But then they went ahead and combined PAC-MAN with the existing CA19-9 test. Why do that if PAC-MAN is already so accurate on its own? I'm a little confused.
It's all about adding another layer of certainty, especially when dealing with something as serious as cancer. Think of it this way. If you get a piece of news from two independent sources, you're naturally more inclined to believe it, right? It's about minimizing uncertainty and boosting our confidence in the results.
Okay, so it's like having two witnesses corroborating the same story. The more evidence, the better.
Exactly. Combining two different tests can enhance the overall accuracy of the diagnosis, providing a more comprehensive picture.
That makes a lot of sense. So given these impressive results, is this combined PAC-MAN and CA 19-9 approach ready for prime time? Could I go to my doctor tomorrow and request this test?
Not just yet, unfortunately. While these early findings are very encouraging, more research is needed to confirm their validity in larger and more diverse populations. We need to see how the test performs in real-world settings before it becomes widely available. It's like a promising new drug that still needs to go through rigorous clinical trials.
Okay, so more research is needed, but let's just assume for a moment that everything checks out in those future trials. Where would PAC-MAN fit into the bigger picture of pancreatic cancer detection? Who would benefit most from this test, would you say?
It has the potential to be a true game changer, especially for individuals considered high risk for developing pancreatic cancer.
Okay, help me out here. What exactly does high risk mean in this context? Who are we talking about specifically?
We're talking about people with a family history of pancreatic cancer, individuals carrying certain genetic mutations that predispose them to the disease, and those with precancerous conditions of the pancreas.
So in essence, for these individuals, instead of waiting for symptoms to show up, which we know is often too late, PAC-MAN could be used to proactively screen them and potentially catch the cancer at a much earlier stage when treatment is more likely to be effective.
Precisely. Early detection is key when it comes to cancer treatment. It gives us the best possible chance of achieving long-term survival.
That makes a lot of sense. It's a proactive rather than reactive approach to cancer care. But what about screening the general population? Could PAC-MAN be used for that as well?
That's a more complicated question. It involves carefully weighing the potential benefits against the potential drawbacks. It's not a simple yes or no answer.
I mean, let's unpack that a little bit. What are some of the potential benefits and downsides of widespread screening for pancreatic cancer?
On the plus side, if we could detect pancreatic cancer early in everyone, it could potentially save a significant number of lives. As we've discussed, early detection is crucial for successful treatment.
But I imagine there's a flip side to that coin, right? What are some of the potential downsides we need to consider?
One major concern is the possibility of false positives. As with any medical test, no test is absolutely perfect. If we start screening everyone, we will inevitably have some people who receive false positive results.
And as we discussed earlier, that could lead to a cascade of unnecessary anxiety, additional testing, and possibly even invasive procedures for people who are perfectly healthy. It's a tough ethical dilemma. How do you balance those risks against the potential benefits of early detection?
It's a delicate balancing act. We have to carefully consider the accuracy of the test, the costs associated with widespread screening, and the effectiveness of the available treatments. It's a multifaceted issue with no easy answers.
So in the case of PAC-MAN, it sounds like we need more data, not just on its accuracy, but also on the long-term outcomes for people who are diagnosed early. We need to know if early detection truly translates into improved survival rates.
You're absolutely right. We need to be able to answer these crucial questions. Does early detection actually lead to better outcomes for patients? Are the treatments we currently have effective at those early stages of the disease? We need this information to make responsible decisions about widespread screening. We can't just jump the gun.
Okay, so it seems we're not quite ready for population-level screening just yet. But there's no doubt that the PAC-MAN test represents a significant step forward in our battle against pancreatic cancer. It's a glimmer of hope, for sure.
Absolutely. This research is incredibly promising, and it offers genuine hope that we can finally turn the tide against this devastating disease. It's a testament to the power of scientific inquiry and innovation.
Hope is a powerful thing. And speaking of hope, let's shift gears for a moment and talk about the broader implications of this research for the future of cancer diagnostics. Where do we go from here? We've covered a lot of ground in this episode. We talked about PAC-MAN, its accuracy, its potential for early detection, and even pondered the idea of population-level screening. But let's zoom out for a moment. What does this research, this PAC-MAN test, tell us about the bigger picture? What does it reveal about the future of cancer diagnostics as a whole? Is this a sign of things to come?
It definitely raises some fascinating questions about the trajectory of the field. The PAC-MAN test, you see, is a prime example of what we call precision medicine, a concept that's been gaining a lot of traction in recent years.
Precision medicine. I've heard that term thrown around quite a bit lately, but I have to admit I'm still a little fuzzy on the specifics. Can you break it down for me? What exactly does it mean?
Well, in a nutshell, precision medicine is all about moving away from that traditional one-size-fits-all approach to healthcare. It's about recognizing that each patient is unique, each cancer is unique, and tailoring treatment to the individual, not the disease.
So instead of using a sledgehammer, we're using a scalpel. We're being more targeted, more precise.
Exactly. Precision medicine is about understanding the specific molecular mechanisms driving each individual's disease. Instead of just blasting cancer cells with the same chemotherapy drugs for everyone, we're trying to, unravel the complexities of the cancer and target those specific pathways that are fueling its growth.
Okay, I see where you're going with this. So how does Pac-Man fit into this personalized approach to cancer care? How does it contribute to this idea of precision medicine?
Well, by measuring the activity of those specific proteases, Pac-Man gives us a much more detailed understanding of what's happening at the molecular level. It's not just about finding cancer cells, it's about going deeper, understanding the processes that are driving their growth and spread, and then tailoring treatment accordingly.
So it's like getting a blueprint, a detailed map of the cancer's inner workings, rather than just a blurry snapshot. We can see the enemy, so to speak, and target our weapons more effectively.
Precisely. And having that blueprint allows us to make more informed decisions about treatment. We can move away from those broad spectrum treatments, which often come with a host of side effects, and focus on those specific pathways that are driving the cancer's growth. It's about hitting the cancer where it hurts, so to speak, with greater precision.
So instead of just hitting the cancer with the same chemo drugs for every patient, we could potentially develop treatments that specifically inhibit those proteases that Pac-Man identifies as being key players in that particular person's cancer.
Exactly. There's a whole field of research dedicated to developing drugs that target specific proteases. And with Pac-Man, we could potentially identify which patients are most likely to benefit from these targeted therapies. It's about personalizing treatment, tailoring it to the individual patient's needs.
So it's not just about early detection. It's about opening up a whole new world of personalized treatment options. That's pretty remarkable if you ask me.
It is, and think about the possibilities beyond pancreatic cancer. This approach, this idea of measuring protease activity, could potentially be applied to other types of cancer as well. It has the potential to revolutionize the way we diagnose and treat cancer across the board.
So in essence, this could be a blueprint, not just for a single disease, but for a whole new way of thinking about cancer diagnostics and treatment in general.
Absolutely. We're moving away from that one-size-fits-all mentality and towards a more personalized, targeted approach, both in how we diagnose cancer and how we treat it. It's a truly exciting time to be in this field. There's so much potential for progress.
It really does feel like we're entering a new era of cancer care, one that's driven by a much deeper understanding of the disease's complexity, coupled with the ability to target it with greater precision. It's a remarkable shift in our thinking.
I couldn't agree more. And that's what makes scientific research so compelling, so essential. It's a never-ending journey of discovery, constantly pushing the boundaries of what we know and what's possible, and who knows what breakthroughs are just around the corner.
Exactly. It's a constant reminder that there's always more to learn, more to explore. Well, as we wrap up our deep dive into the fascinating world of Pac-Man and protease activity, what would you say is the most important takeaway for our listeners today? What's that one key message you'd like them to remember?
I think the most crucial takeaway is this. The future of cancer diagnostics isn't just about finding those cancer cells. It's about delving deeper. It's about understanding the intricate dance of molecules, the underlying biological mechanisms that drive the disease, and using that knowledge to develop more personalized, more effective treatments. It's about giving people the best fighting chance we can.
Beautifully said. And on that note of hope and progress, we wrap up another deep dive. Thanks for joining us today. We'll see you next time. Keep those brains buzzing.